for the morning, and it's going to follow this afternoon by what we call a wrap-up session. We'll see how it goes. Where we get together and discuss what happened this week. And we'll hear talks from some of our friends who haven't spoken yet. So please come. That's 1:30 to whenever. And now it's a great pleasure to introduce Carl Bergstrom from the who is going to tell us about defensive complexity in coevolutionary arms races.
All right, well, thank you very much. It's a pleasure to be here. And what I want to do today is talk about really talk about a problem rather than a solution. It's a problem that I think biologists could benefit enormously from the help of computer scientists, and computer scientists, if they haven't completely solved it already, might find extremely interesting to think about. So this is we're going to talk about sort of this problem in understanding immune systems. And we'll come to that. One of the things that's kind of surprised me here in a very pleasant way is the degree to which the evolution of evolvability has kind of come to the fore as something that people are very interested in. I've seen a bunch of common themes there, a bunch of interesting mechanisms, and it's gotten me excited about that. That's definitely one of the things I'm taking back to Seattle with me, is kind of renewed excitement there. What I've been interested in and what I'm going to talk about today is sort of the flip side. I want to talk about the evolution of unevolvability, and in particular, how does one species make it difficult for another to evolve? So in biology, we spend a lot of time thinking about how do you win evolutionary arms races, and sort of the Red Queen, the kind of Red Queen dynamics are our sort of standard way of thinking about evolutionary arms races. Each species tries to evolve faster to keep ahead of the other one. You have to run as fast as you can simply to stand still. And so there's a tremendous amount of work that's been done on looking at the dynamics of these kinds of coevolutionary interactions and looking at how you win an arms race. There's been very little work done on a second aspect. The other way to win an evolutionary arms race, instead of being a fast runner, is to find a way to force your competitor to evolve slowly. And so that's what I want to explore today and talk about a little bit. Like a lot of great ideas in biology, and game theory and economics and so forth, I think this one really, as best as I can tell, can be tracked at least back to John Nash. John Nash was, in the 1950s, very interested in the arms race. And this is probably, I'd be curious if this is kind of part of the story that computer scientists all know when they think about these things, or whether Nash's contributions are less well known. But anyway, I'll tell the story briefly. Nash was very concerned with the race between code makers and code breakers. It was between enciphering and deciphering. And this was a very pressing issue for national security. It was just a few years after Turing and the Enigma machine at Bletchley Park, shown here, and so on. And what Nash was interested in was, exploring the mathematics underlying encryption. I feel a little silly saying this to computer scientists who know these things much better, but Nash wanted to know, can any code be cracked, and how, and can one make an uncrackable code? And if so, how do you make an uncrackable code? And so in the 1950s, Nash worked through the mathematics of all of this and then sent a remarkable series of letters to the National Security Agency explaining his discoveries. And so they were just declassified last year, and so here's the start of one of his letters, and you can kind of see, you can kind of imagine what it would be like to receive a letter like this. It's many pages long with schematic diagrams of machines and so forth. And so in this, he explores the problem of how hard is it to crack any code. And he writes something that, given my knowledge of the history, is, I think, quite remarkable for 1955. He says, so he's interested in this code cracking problem, and he realizes that actually this can be made arbitrarily hard. He says, for all sufficiently complex types of enciphering, especially where the instructions given by different portions of the key interact complexly with each other in determination of the ultimate effects on the enciphering, the mean key computational length increases exponentially with the length of the key. So that's 1955. And then the other part of the story that I very much like is that then he sees immediately where this is going to go in terms of the coevolutionary arms race. He says as ciphers become more sophisticated, the game of cipher breaking by skilled teams, etc., should become a thing of the past, because you can make these codes exponentially hard to break. The arms race between enciphering and deciphering is going to grind to a halt with a decisive win for the encipherers. So he sent this to the National Security Agency, and they, read through it, concluded his system didn't meet their necessary security requirements, informed him that the reply is written and they're not interested in further coordination. So at the time, it wasn't properly appreciated. But I think it's a deep insight, and I'd love to hear, in the questions about more of the history of the other ideas that were floating around at the time. What I think, fundamentally, to try to capture what it was he was saying, is this idea that I'm going to call defensive complexity for now. The idea that a mechanism could be made secure against subversion by using intractability or time complexity of learning as a defense. So if I deploy some kind of a system, I don't have to necessarily prevent you from accessing it as long as I can prevent you from figuring out how to operate it effectively. So and we could take this into sort of the biological domain. It's kind of rephrasing this in the biological domain. I'd say a system manifests defensive complexity if the adversary population will take a long time to evolve to subvert it. So now the main change here, I'm specifying a particular learning mechanism, namely evolution by natural selection, as a possible way, as sort of the way that I want to look at the adversary population doing its learning process. So when we go now, let's kind of return to coevolutionary arms races, and I want to focus for much of the rest of the discussion on the problem of coevolution of hosts and pathogens. And I find it absolutely amazing that we're actually here to have this conversation. I think it's incredible, nearly incredible, that large, long-lived multicellulars can make a living in a world where pathogens are able to enter their bodies, enter their cells, subvert their gene expression, and so on. So how do you win? It can't be straight red queen dynamics that allow us to keep up in this arms race. The generation time of viruses is approximately 100,000 times ours. They're very have very, very large population sizes. We put them under intense selection with the immune systems we deploy. And so I want to try to ask, how can an immune system possibly work against an adversary that has that enormous evolutionary advantage? And so, of course, deploying the immune system and possibly sex against parasites and the running an evolutionary process sort of ongoing within the immune system may be an important part of how we do this. But if you kind of think about the task that an immune system has to face, it has to perform in order to function well, you need to build a system that's highly sensitive. You need to respond early to infection before the infection does too much damage to the self. It needs enormous breadth. It needs to recognize any new pathogen. It's not good enough to have immune systems that are responsive to 6 common pathogens, but you have to be able to respond to anything that possibly could come along, things that perhaps your parents never saw. You need learning. You need to be able to distinguish self and non-self. I think this is sort of critically important in making immune systems work is the fact that we can't encode, I believe we can't encode genetically what is self and non-self in any kind of obvious way because of sexual recombination. And so we can't have coevolution of a set of safe MHC molecules, say, with our own proteins, because we're going to get a whole new set of genetic variation coming in from the other parent. So this has to be done in a learning fashion. You need high specificity. Once your immune system starts to respond to things, it's no good clearing the flu if you take out your liver in the process. You need to be able to generate a very large-scale response. You need sort of massive amplification, where you go from a very small number of cells in the repertoire up to a very large number in an immune response. You need coordination. You've got this sort of, you're building, when you mount an immune response, you're building a distributed tissue in your body, and that tissue needs to be coordinated in its development and in its behavior and function. And ideally, well, we're wishing, why not make good use of prior experience? Why not try to develop some kind of memory? This is, I think, an enormous challenge to evolve a system that can do all these things. And there's sort of two additional challenges as well that we face, and one I've sort of already alluded to is we've got an immune system has to avoid autoimmunity. Immune systems can't afford false positives, and strictly speaking, it's not so much that they avoid all false positives, but they are able to turn off the immune responses that are launched as a result of false positives. And at the same time, immune systems have to avoid subversion. Pathogens have evolved to sneak under immune surveillance, to disrupt the immune function, and to use whatever mechanism possible to try to escape the effects of the immune system. I'll jump. So there's a sort of a dual challenge for immune function. The goal of the immune system, you might say, is to reduce damage from pathogens, and to do that, it's got to avoid immunity on one hand. It's got to avoid subversion on the other. And the reason this is particularly tricky is I think these are in fundamental opposition. What you need to do to avoid autoimmunity is you need to have ways of shutting down an immune reaction. You can't possibly prevent all false positives over the lifetime of the organism. And so, in order to avoid these autoimmune reactions, you need control mechanisms that disable the immune system. Of course, if you want to avoid subversion, sort of the worst thing you could do would be build in an off switch, because that would be the first thing that pathogens would evolve to subvert. I'll just jump back here. I had those slightly out of order. So what I wanted to mention, many vertebrate viruses do try in various ways to subvert the immune system. The pox viruses make a living this way. They tamper with the immune regulatory signals. They produce proteins that stimulate the immune receptors. They do sort of everything you could imagine. They affect the function of immune cells and their own signal production. They produce decoy receptors that they kind of flood the body with decoy receptors that mop up the signals so the signals can't reach the true receptors. They have other chemicals that block the cytokine receptors. They sabotage chemokine signals in various ways. They have proteins that slice up our immune signaling molecules. So you do all of these sorts of things, and nevertheless, they're not particularly good at it or particularly subtle. Pox viruses hijack an airplane by running into the cockpit with a hammer and swinging it around and around and around. They don't fly the system with any kind of level of subtlety or control to actually work the system to their own benefit. They're not as elegant, say, as some plant viruses, viroids, are in dealing with a much less sophisticated plant immune system. The RNA silencing system serves as a plant immune system, and other organisms as well, but it's conserved in plants. And there are these plant viruses that will come in, and what they do, they express a little RNA that in double-stranded form, so that it triggers the RNA silencing system. That RNA is identical to a critical component of the system itself. So they come in, they express in double-stranded form an mRNA that then causes the plant RNA silencing system to recognize itself as a target and disable itself. So they're able to come in and very precisely knock out the immune function of plant cells. Okay, so I'll just jump forward here. So, I mean, the problem that I want to kind of think about, and the problem that I would like help solving is that communication is absolutely essential for the immune response. We need to be able to shut down unwanted reactions, but communication can be subverted. As I learned yesterday, there was actually a class of problems that I think I need to learn quite a bit more about, these sort of Byzantine generals problems that I guess if I'd had an undergraduate education in computer science, I'd know quite well. But I think the immune systems are dealing with quite a kind of Byzantine fault tolerance issue, in that there is no kind of master parliament that can vote and count up, votes from the various immune cells. They have to do all of their signaling in these pairwise fashions, where some fraction of the immune cells are quite possibly infected with virus, and therefore have their gene expression massively disrupted, and so are acting essentially as traitor generals. I don't know. I mean, I don't know exactly what the most useful way to kind of leverage all of this literature to think about the immune problem is, but I was excited to learn about it, and look forward to reading up on all of that. Let's see. So I can. This is just sort of an attempt to think about this type of problem. We can think about the distinction between robustness. Robustness would be just to throw out a definition of preserving phenotype or function despite variation in environmental conditions, noise, and component failures. And I don't know if this is the best term, but this is the one we ended up using. As we kind of contrast this to strategic robustness, this is sort of an average case sort of a situation. We contrast this with strategic robustness as how do you preserve phenotype despite directed attempts at sabotaging the system. This is sort of a worst case robustness or tolerance, if you will. And so what I want to figure out is how is strategic robustness manifested in the immune system. So we've got this puzzle. Why don't pathogens evolve to simply shut off immune responses? And the hypothesis I want to propose, and hopefully I'll have a little bit of time to discuss with you, is that perhaps immune systems do this because their control logic, the kind of the logic of the cytokine signaling network, is defensively complex. It's sufficiently hard that pathogens are not going to easily be able to evolve to exploit it. I think there's some kind of intriguing biological observations that make one think in this direction. One thing that puzzles me quite a bit is that some mice and men diverged 100 million years ago, and yet their cytokine signaling networks are strongly conserved. So the basic fundamental signaling pathways between mice and men are almost exactly the same. That is, we've learned almost everything that we've learned, not almost everything, but we sort of, the way that we start to try to make sense of immune function is to look at immune function in mice, and through that we're able to build up these complex networks of different cell types signaling by various interleukins and other cytokines. And this whole structure has been conserved for 100 million years down two branches. So you've got 200 million years of evolutionary divergence on a structure that should be locked into the most intense coevolutionary arms race that we're experiencing at all. Why don't we see signature? Why don't we see change there? Why don't we see strong selection? So you can.
Say conserved, you find it also in human? Why you say conserved?
Yeah, the basic structural.
Same
Very much, very similar. Yes. So the structure of the signaling networks are very much the same, to the point that we can, as a first approximation, do immunology by. Immunology is described as sometimes as the interaction of mouse and bacterium mediated by a hypodermic needle, and that's how we study immune systems. So we can look at we can look at selection in various ways, and most cytokines show the opposite of selection in the kind of ongoing evolutionary arms race. They show purifying selection. They're highly conserved, in genome-wide analysis. And this is the opposite of what we see in the immune effector cells, the cells that actually immediately touch and mediate and interact with and kill. And immune systems are under strong positive selection. So we see massive sort of selection for divergence, for difference, for fixing new variants in the sort of the front line cells. But in terms of the control machinery in the back, that's all very strongly conserved. And I think that's an interesting puzzle. And it's interesting to think about whether this kind of defensive complexity story has something to do with what's going on. So,
that's not a key, because you can break each part of the cytokine system one at a time.
Well, if you break it one at a time, the system tends to get even more ramped up in some sense, right? So it's so it's very hard to it is very much like a key, I think. Would be my proposal. I expect that's one thing we need to understand better. But there's this type. If we go back to go back to my diagram there. Knocking out single interleukins will not necessarily disable the system. It may actually amplify the ensuing response. So that's an interesting observation. So how do we model this story? I'm coming at this as a game theorist, and maybe this isn't the best way to approach the problem, but I'm going to lay out one kind of way of thinking about the problem, and hopefully we can talk about some others. I want to think about this as a signaling game. There's this huge literature on evolution of signaling games, and the basic idea is that you've got Alice gets some private information about the world. Bob has to choose some actions, and so Alice picks a message from some message set, sends it to Bob. Bob chooses actions. We're thinking about subversion here, and so what we want to think about now is we want to add another player, Eve, who now steps in and not only eavesdrops on the message, but can actively tamper with it and change it to anything else that she wants to. So you can try to make game theoretic models like this. I think they're very interesting. One of the big challenges in doing this is trying to circumscribe the set of things that Eve is allowed to do. At first glance, if you allow Eve to do whatever she wants, then it might be impossible for Alice and Bob to communicate.
So could you just say, in the immune system, who's Alice, who's Bob?
I will definitely do that. I'll definitely do that. So I just want to kind of work through the logic of why I'm using the model I do, and then people may have other very interesting ideas. So we've got Alice communicating to Bob. Eve can send some kind of message. Eve can tamper with the message. And if she can do whatever she wants, we think that maybe Alice and Bob can't communicate. However, if Eve can't figure out the ways to manipulate the signal in order to confuse Bob, then signaling may actually be possible despite Eve's ability to interfere. So when I want to go from this kind of situation. This is a little bit hard to model in game theoretic context. We've got 3 players. That already makes things quite messy.
Carl, I must stop you here because I'm losing because it refers to what you said for why I could not follow the arguments leading to why communication, and now you build on this. Why communication is necessary here? What is communication? Wait a minute. And what Alice and Bob represent?
I will tell you in a moment what I'm going to do with Alice and Bob. Communication. I'll just tell you right out. Alice and Bob are both components of self, right? This is the immune system. The reason communication is necessary is because to generate this massive distributed tissue that performs a specific function, you need coordination. Without communication, very hard to confer the necessary coordination in function and behavior. So, if it were possible to simply send off kind of prime the immune cells, send them off, and let them all follow entirely pre-planned progression programs, maybe one could dispense with coordination. That doesn't seem to be how the immune system does it. And I think you could sort of think about a lot of the problems that would involve, that would arise if you couldn't allow immune cells to communicate at all after a reaction is launched. So that's why I think communication is so important. Thank you, because I should have made that clearer as I was discussing all of this. So what I want to do is kind of turn this into a signal tampering game that we can, this is a signal tampering game now, I want to turn it into one that we can use to think about the immune system. And so I want to go down to 2 players. And I want to think of internal communication among the systems of one player, right? So Alice and Bob are both the immune system, different parts of the immune system, communicating in order to coordinate their response. So we have 2 players in our signal tampering game. We've got a host who processes information, and we've got a pathogen who tampers with signals. And I have this kind of schematic that I've used to think about it. I think about a host, so what happens when a host processes information? Well, there's some kind of cue in the external world, or maybe within the host's body, goes through some black box and generates a response. What's in the black box? Well, usually some kind of sensor that is involved in detecting the cue. The sensor is going to take this external cue and transduce it into internal signals. These might be the cytokine signaling molecules. They might be various other elements of say T-cell activation. And then there's some control logic that maps from the internal set of signals, the, say, concentration, abstractly, the concentrations of the cytokines or whatever the internal signals are. It maps from that to the response. So the second player in my story is a pathogen. Yes?
So a computer scientist would maybe say that control logic or the signals also change the state of the box.
Right. I was thinking about, and maybe I should have something like this. I was thinking about so I was thinking that the signals were the tape, so that all of the state changes are manifest by settings to the internal signals. So the signals need not be a sort of a one-time neural impulse running from here to there. They can be the concentration and the location of a particular signaling molecule.
So you have some vast combinatorial collection of possible.
That was sort of what I was thinking, is that this would encode. Yeah, is that this was going to encode the kind of sum of the knowledge of the system at any given time. When we, in our original crack at modeling this, these actually were chemicals that were evolving according to a set of differential equations, and that turned out to be messy and so on. But that's kind of how we were thinking about it. But tell me, I mean, I definitely want to figure out how to. I think there are some nice things about modeling it this way, but it also there's some distance left to go. So we've got a pathogen. The pathogen has some kind of tampering strategy by which it can tamper with the signals, so it can modify the signals. My idea, here is we'll think about the pathogens not able to change the cue. The cue could be, for example, the presence of the pathogen within the host's body. Can't directly impact the sensor, but can do all kinds of things to the signals, or equivalently sort of memory of the system, and thus change the response generated by the control logic. So in the game theory view of it, the hosts, what I allow the host to do is to choose a control logic. So the host gets to choose a mapping going from different combinations of signals or different levels of signals to the response, and the pathogen then gets to choose a tampering strategy. So what I'm going to do to give the pathogen as much of an advantage as we can to kind of represent the fact that once inside viruses are able to, once inside an immune cell, control expression by that cell, but they're also able to pick up genes from the host genome. I'm going to make what I call the universal constructor assumption. Simply means that the pathogen can make any signal used by the host, so it can produce signals, it can up or down regulate them, but doesn't know what the signals do. and doesn't know how the signal perturbations map to responses. So, to try to build...
Specifically pathogens affecting the immune system.
Affecting the immune system, yes.
Infecting directly.
I think that may be one particularly effective case, but certainly not necessary. They could also they can infect other tissues and generate cytokine signals from the genome of the host, even if it's not necessarily cytokine producing. So I think there are a number of possibilities. Okay, so just to try to fill this in a little bit more, let's think about control logic that maps from signal values S1, S2, and so on to some scalar response R. And the pathogen then can alter each signal by upregulating it, downregulating it, or leaving it untouched. We can assume that sort of the host's control system is making whatever decisions it needs to in ways that would be optimal if untampered with by the pathogen. So the host is best off with the default default response generated by the unperturbed signals, and then we can think about how the response affects the pathogen. We could imagine the response as some aspect of immune reaction or sensitivity, and so stronger responses correspond to higher values of R, acting as more harmful to the pathogen. So what I could imagine is R unchanged is the best for the host, medium for the pathogen. R increased is bad for the host. It's bad for the host to be moved away from its own default ideal settings, but it's also bad for the pathogen. If the pathogen messes up and sort of cranks up the immune response instead of cranking it down. R decreased is bad for the host, good for the pathogen there. It's sort of cranking down our immune response. And the sort of key insight that I think for me in thinking about this was just to recognize that the control logic that the host deploys induces a fitness landscape on which the pathogen evolves. So I want to think here about this sort of situation in which the pathogen is evolving, tens or hundreds of thousands of times faster than the host. And so, what happens here, basically the host is going to make a single move, pick a control logic, then the pathogen will actually get to take a whole adaptive walk on this landscape. And so what the host is really doing is picking the fitness landscape on which the pathogen gets to proceed. Yeah?
So is it reasonable to think of the host response as one-dimensional? You're sort of using this R as a quantity that goes up or down, and that sounds a little bit limiting.
I'm sure it's very limiting. I think it's I wanted to try it as a first pass to think about how this situation might work. Ultimately, when you think about these things in game theoretic context, I was sort of thinking, well, ultimately there's a payoff to the host and a payoff to the pathogen, and these are scalars, and individuals are trying to maximize their scalar payoffs. And so I was thinking, R could be, we could map payoff space to R or something like that. But yeah, sure, it'd be interesting to think about controllers that control more than one axis.
But from an engineering point of view, that seems to be one of the best strategies. If you want to send a simple message reliably, you utilize multiple pathways, and then it's very difficult to obstruct them all. And what that corresponds to is increasing the dimension in which the space in which this fitness landscape exists. Because if you disrupt one signal, you get very little feedback that you've accomplished anything, because so many other signals are coming through.
So I pretty much don't need to finish the talk at this point, because that's going to be exactly my main point. Is by increasing the dimensionality of the signal space, you can make this landscape extremely hard for the pathogen to evolve upon. So I think you're saying.
My apologies.
No, well, that's great. It means that something I said is making sense, which is gratifying as a speaker. Okay, so at this point, I throw in a bunch of machinery. I'm going to go through it quickly, and I don't think the machinery is fundamental to what I'm talking about here. It's just an example of the kind of thing you can do. What I want to do is I want to model this control system. I'm going to do it as a ternary logic. so that we can easily think about perturbations up, perturbations down, or leaving things constant. And then we can think about, the host gets to specify its control logic in this in a form like this. So if S1 is increased, XOR S2 is not increased, then R is increased by 2m. Remember, that's the worst thing for the pathogen, so that's bad for the pathogen, and we can kind of read these conditions off. You can read these off, and so that logic there, if you went through and checked it, builds a fitness ramp like this, a sort of a very simple fitness landscape that a pathogen could evolve on. We could build a more challenging fitness landscape. Here's kind of a checkerboard pattern where you start here. Here's the unperturbed state. Any single move by the pathogen makes things worse for the pathogen, but it does have this target it could reach over here. So, and so one could kind of think about how different fitness landscapes affect the rate of the evolutionary process. This is kind of fun, without knowing who was going to be here or anything. I used these two as examples in the paper. This fitness landscape is very easy to learn. There's a result by V. Bergman, who's here somewhere, that this takes time proportional to the square, 1 over the square root of the population size. So it's very easy to learn this. Whereas this fitness landscape, this checkerboard using a result from Dan Weissman and Daniel Fisher, it takes time that's exponential. And this is the stochastic sequential fixation domain, if I remember right. Actually, if the population is big enough, you do go through in the deterministic domain, and so on. So we can take these kinds of results from population genetics and use them to figure out how hard it is for the pathogen to be able to exploit this control logic. Then, sort of the key theorem that we could show, and in some ways this is sort of obvious, based on what you said, is the host can construct control logics that require time that's exponential in circuit complexity for the pathogen to learn to exploit by natural selection. So the host can deploy arbitrarily, sort of circuit complexity in, the host can deploy a control logic that takes time e to the n to learn. The proof is by construction, the easy kind instead of the hard kind. And the basic logic is, one could do any number of different alternatives. I wanted one where there were sort of increasing number of targets for the host as you increase the dimensionality. It's I'll call it peaks on corners control logic. Here it's written out. We could go through that if we had more time. And the idea is, in 2 dimensions, the pathogen benefits if it can get to the corner, but it has to go through these valleys. 3 dimensions, it would look sort of like a Rubik's Cube like this, and you'd start at the center, and now you have to take 1, 2, 3 steps in the valley in order to get out. And as we move up in dimensions, just as you said, this thing becomes very, very hard for a pathogen to traverse. So this is a very, it's simple to actually write down the control logic. The sort of the length of the control logic requires order n operators to specify for an n-dimensional system. But it's very, very hard for a pathogen to move through. So we can prove that the time a finite pathogen population takes exponential time to cross a fitness valley of n steps. And so that's just a little lemma that you can show. It's kind of intuitive and slightly harder to show than you'd think it would be. And so, this gives us the result. We build this n-dimensional peaks on corners control logic. So you use a linear number of gates to make this fitness landscape that takes exponential time, and then that proves the theorem, because the theorem is, just simply says it's doable, and we provided the construction. So, sort of that's the basic intuition. I think that could probably be done more elegantly, but I wanted to just give you a sense of the kind of way you can try to model the problem formally. Where this leads me to is to come back to one of the questions that got me started on this. When I was a postdoc, I had an extra year between when my job started and where I was, and kind of wrapping up some projects, and I decided to spend the year learning as much as I could about immunology with Rustamantia. And this is the main thing I learned. Everything I learned in 2000 is now wrong, but even if it wasn't wrong, I've forgotten it all, because immunology is spectacularly complicated. And so, why is immunology so darn complicated? Well, if pre-meds could learn it, pathogens could too. And so, anyway, that's the other thing that I think is you notice I didn't talk at all about how defensive complexity evolves. So I talked about the evolutionary consequences of it, but I haven't talked about what mechanisms lead to its evolution. And so we're talking about the evolution of evolvability. I think it would also be very interesting to think about the evolution of unevolvability. Yeah, so that's what I have to say. Thank you.
That's really interesting ideas there. So you showed the diagram of Alice needs to communicate with Bob in a way that Eve can't intercept or interfere with, right? And you showed that Alice can make up a code that only takes a polynomial number of
linear, in fact.
Sorry, the complexity of the code, cracking
right,
is exponential in the number of
the complexity of cracking is exponential, yes. And I'm lucky here because I know the learning algorithm, so I don't have to do all learning algorithms. I can use natural selection.
How's Bob going to crack it? Right, Bob needs to be in on the code too, right?
Right. Well, that's
Sorry, the question?
So how does Bob crack the code? Well, presumably there's advanced coordination that's possible between Alice and Bob. Alice and Bob are, of course, the same organism, and this is your evolution of unevolvability problem. How did the immune system manage to evolve a cytokine signaling network that's complicated enough that it's uncrackable, yet it was built step by step by step in order to have that complexity functioning all the way along the way. I think that's the next area that I want to try to think about.
Two questions. One is if you said the structure of the system, logic is a constant by and large between mice and men. Then the pathogens actually don't need to. They could kind of like have information over longer time steps, right?
That's true. It doesn't have to be done within the evolutionary time of a single individual.
So you need to vary something within it, otherwise the pathogens will eventually, find not over a single host lifespan, but rather.
So I think they won't. I think if it's exponentially hard, it doesn't matter whether you give them one host lifespan or a polynomial number of host lifespans. Either way, they're not going to crack it.
I see. Just another question. I wonder whether there's, kind of like some logic predictions that you could go and look at the wiring of the immune system to see whether it's indeed rugged and high dimensional. And
I think that would be a great thing to do if you could figure out how to, to try to actually construct what does that fitness landscape for perturbations on the cytokines look like. That would be a really neat thing to check out. Haven't even tried to do that yet. The only predictions that we've sort of managed to check were simply the ones that you'd have the cytokine signaling network under stabilizing selection, but the effector molecule is under positive selection, and that turns out to be the case.
So since you used the language of cryptography for a lot of this, there's also a further thought. If the control logic is a one-way function,
yes,
meaning that if a virus desires a certain response, it doesn't know how to tamper with the signal to produce that response. That's the generalization of the particular situation you're considering.
Okay.
And it could be robust against viruses or pathogens that can evolve in a more sophisticated manner than you're modeling right now. I mean, so maybe they don't have to make one change at a time and have difficulty getting to the corners as you have here. Maybe they're capable of more sophisticated evolution in some fashion.
And it could still be.
It'd still be secure if you had a one-way function of a suitable capability.
No, I suspect. I think that's right. And one of the things I think would be very interesting to kind of look at. How hard is it to evolve one-way functions? How hard is it? Basically, let's start. I've just done the consequences. Let's start talking about how you actually get there. I think it'd be really interesting. Nick.
So it seems at least as hard to demonstrate that these systems have evolved to be unevolvable as to demonstrate evolvability. So the point is that
Yeah.
suppose that the immune system is very complex and it's evolved this very rugged fitness landscape from the point of view of the pathogen, which it turns out makes it hard for a pathogen to crack the code. But how could you go about showing that that is an adaptation rather than just immunology is complicated?
Right. So this is something I've worried about. If it were a matter of, I think basically one would want to look at the consequences of perturbation. So if it were a matter of immunology being complicated, which I think is, should be a null hypothesis in this case. If you had a Rube Goldberg machine instead of an uncrackable code, it should be that almost anything you do to it breaks it, rather than sort of amplifying the response. And the immune system seems to be the other way around.
So
That's my best, the best thing I've got so far.
You can sort of see a glimmer of some predictions, but pretty tough to test.
I think so. Yeah, exactly. Yeah, and that's one of the things that would be I'd like to learn more both on the. I mean, you see, I'm just the conduit between Eric Chastain, who's the computer scientist, and Rustamantia, the immunologist, and so I'm just up here, kind of making up stuff on both sides of the aisle.
I think it's a very nice idea. I'm a bit confused by how the communication from A to B can be robust to mutations if the fitness landscape is so rugged. So how does the system deal with?
That's a very good question. That's a question I need to spend some time thinking about. That's a serious concern. It's part of this evolution of unevolvability puzzle. Is that how can you have a system that can continue to add complexity when complexity, when perturbations have this generally deleterious effect? I think, yeah. I'll leave it at that because I don't have a better answer.
But you've observed, with your construction, that you only need to add a fixed number of gates to the
That's
encoding
right.
network to make it a geometric factor harder for the adversary. So
That's right. I mean, you have to do fewer steps. I'd love to have a better explanation for why those steps were even evolutionarily easy to make.
But you could say that it has to be harder for the host to evolve the system than for the pathogen to outwit it, because of the fitness landscape, which is that any change is bad from the host point of view, whereas half the changes are good and half are bad for the pathogen's point of view.
So, one might hope that you'd be able to step. Yeah, that's a major concern. Let's just.
the moves in the host and the pathogen see different fitness landscapes because their moves are different.
Yeah,
The
the
host
geometry
moves
has to be different.
are adding gates to the
Yeah.
complexity of the encoder.
Yes.
And the pathogens moves are navigating the fitness landscape encoded.
Although the way you set it up, they were the same. They were the SIs, but maybe there does have to
Well,
be
I think the host,
different
the host is at a working position. So what is the host? What's the main move the host needs to make? Just add one more signal. And so right now you're in the center of the hypercube, and now you add one more signal, and all you've got to do is get that signal level right, because everything else works right, and you just add in this signal so that it interacts complexly with some of the other signals and has a proper level. So you're kind of doing these linear things. You're not doing these hard explorations. And then the other thing that's useful, of course, is there is individual selection for getting this right, because you do have 100,000 generations of bacterial evolution, of viral evolution in your body, and so not only is this beneficial at the kind of lineage level, but is even beneficial getting it right yourself. Chris?
Continue what Nick started. I think that the landscapes are different, and in fact, in some sense, the Rubik's cube, the n-dimensional Rubik's cube, is the global maximum. Could be. I conjecture, but there could be other. The question is, are most hills are really bad?
That's a good question. And my intuitions about the geometry of these high-dimensional spaces are terrible, and so I don't even have a guess.
Now you are talking about a space where every point is topology of this high-dimensional space.
So if you wanted a mechanism that cheaply generated this encoding strategy, it seems that you'd want a gene duplication to create a copy of an existing signal, and then mutation away from that, and every time you moved away from it, changed the nature of the signal that was being used, you would immediately see an improvement because you'd be making, you'd gradually raise in these extra.
That's a very nice mechanism.
Spikes in the landscapes you might have.
Okay.
I'm just wondering though, the pushing of adding levels to the host is pushed by the selection pressure from the pathogens, right? So
yes, I think so.
Essentially, you kind of like expect the host to be like one step away, no more than one step away from where the community of pathogens have reached or something.
You might expect something like that. Yeah, there's a nice cancer suppressor analogy, I think, there.
It also looks like the signal integration part would have to be somehow highly delocalized from the point of view of the host. Because if it were localized, then there would be a point at which the virus could just kill the whole system.
Yeah, that's right. I mean, I think this definitely has to be done in a highly distributed way. That's part of why I got quite excited about this Byzantine generals story, because it is so massively delocalized. Yeah. All right. Well, thank you all very much. I appreciate it.